Point 2, it is immunities and the blood type. Okay, the immunities means in Thai we use the term ภูมิคุ้มกัน it is the how body responds to the germs, and we have many type of germs. For example, the virus, the bacteria, the fungi, and the parasite. Or for example, it is parasitic worm. Like this. So we can classify the type of the immunities here into two types. The first one we call first line defense. For the first line defense means a situation that you have the external body, for example, the skin and the sweat or even the tears. The skin here can use for the protect the internal organ. So we can call this one as a protective layer. And the sweat and the tear, it is liquid chemicals inside the body. So the sweat and tear here contain some chemicals that can destroy the germs. Okay, and the second type we call the second line defense. The second line defense is the chemicals and the cells inside your body respond to the germs specifically to deal with it. And for example, it is the interferon. The interferon is a kind of chemical used for deal with the infection of the bacteria and the fungi like this, or even the virus. And the immunoglobulin, this one here in short we call antibody. Okay, the antibodies. And we have the white blood cells. The white blood cell is a cell that take responsibilities on the production of the immunoglobulin or the antibodies, or even produce the eating process of the germ cells. Okay, the immunities here is a response of the body mechanisms to control the invasive organism or the germs, okay, as I mentioned earlier. And the example is a bacteria, virus, protozoas, fungi, and the worm, like this. And the two processes that we deal with the germ, it is the phagocytosis. The phagocytosis means the process that immune cells or the white blood cells here eat the germ by producing the part of the body here to wrap the germ cells. This is the germ. Okay, and this is white blood cells. And the center here, it is a nucleus. Okay, and we have the type of the white blood cell that deal with this one is the neutrophils and monocyte. Okay, and immunization, this process is process that use the immune cells here produce the specific recognized to the germ. Okay, for example, this one here, they produce the molecules. This one is the antibody. This one is the white blood cells produce the antibody or the immunoglobulin, okay, to bind with the surface of the germ. Okay, the molecule here, the blue one here, will bind with the germ. This one, as a red one here. When they bind it, it will be eaten by the neutrophil and monocyte by the phagocytosis here, easier. Okay, and the time to respond for the germ here is more quicker than the phagocytosis only. And the next time here we talk about the white blood cell staining. Basically, white blood cell cannot be seen, okay, under the microscope because the cell is quite clear. So we need the staining method เอาการย้อมสี and we use many different type of dye or the colors. Okay, we group them into three groups. The first one, the dye that dissolve in the acid. If you use the dye to dissolve in the acid to stain the white blood cell, that those white blood cells we call acidophile, because the word phi here means love. They love the acid. And the dye that dissolve into the base we call basophil. Okay, the white blood cell that stained by the dye that dissolve in the base we call basophil. And if the dye dissolve in neutral pH, this one here, and you use this dye to stain with the white blood cell, we will call this one as a neutrophil. Okay, and the dye dissolved in the acid we call acidophil. And the example of the red colors that we use for staining, in general staining of the animal tissue, the name it is eosin. So we can call eosinophil. okay, and a dye that dissolves in a neutral solution, this one we call neutrophil, okay, and the dye that dissolves in the base solution we call basophil, like this. And the other characters of the white blood cell here, you will see the dot inside the cells. The dot here refer to the proteins, and the protein basically it is the immunoglobulin or the antibody. So we have two types, the cell with dot and the cell without dot. If you see the dot, we call granulocyte. The word dot, the granule here mean the dot, so we call granulocyte. And a granulocyte for the cell that you did not see the dot. And we have many types of white blood cells or the leukocyte. The first one is a neutrophil. Neutrophil here, the nucleus contain many lobe or pool here. This one you will see three lobe for the neutrophils. Okay, and you can see the dot, small pink dot. Even the dot is quite different. Sometimes you can see a very clear dot. Sometimes the dot is not clear, but it is dot. But sometimes you cannot see the dot like this. So it's red and blue granules. And the eosinophil here you can see two lobe nucleus, and they have a red granules inside this. And the basophil here you see two lobe, but this picture you cannot see the lobe because the granule is quite large. Sorry. So when the granule get quite large, I think you cannot see the nucleus, but they say that it is too low. And you can see the purplish back granules สีม่วงออกดำ and for the agranulocytes, this one is lymphocyte and monocyte. They have basically they have round shaped nucleus, and they have very thin layers of the cytoplasm, and you have pale blue cytoplasm. And the monocyte here will be kidney-like. Nucleus look like kidney-like like this. Okay, and they have pale blue cytoplasm. So this is characters of 5 important white blood cells you're supposed to know. And this is process of phagocytosis. Okay, first step you have neutrophil. The neutrophil here rolling, and neutrophil is inside the inside the blood vessels, and they get rolling and attached to the surface here by the protein. Okay, and after that the cell here become adhere to the surface and become flattened, spreading like this, and they enter into the tissue. Okay, pass through the walls of the blood vessels, and they move toward the bacteria because the bacteria produce some chemicals. Okay, and the white blood cell itself also produce some chemical. This one we call chemokine. The chemokine here we call the other bacteria, the other white blood cells here come into the site of infection, and after that the white blood cell here will start to eat the bacteria. So this one here we don't have the immunization process. It just only did phagocytosis only. And the immunization here we can classify into two types. Okay, the first one is active immunization, and the second one is passive immunization. For the active immunization take roles on the production of the antibody by themselves. The white blood cell will produce the antibody by themselves, but the passive is quite different. This one here they receive the antibody from somewhere else. Okay, that is not by the process of the body of the white blood cell to produce it. And the example here of the active immunization is vaccination and the direct contact with the germ. Okay, you can develop the antibody by yourself. And the passive immunization, this one you take the serum or the serum. In Thai we use the term serum, okay, but this one here serum. And colostrum. Colostrum means the clear, like when the mother take the breastfeeding to the baby, we have two types of the milk. Okay, the first one is like a liquid, the liquid, and is quite white color. Okay, and the other one is clear colors. The one that get clear colors we call colostrum or น้ำนมเหลือง. And inside the body here of the mother here we also have the placentas, and the placenta here also exchange the chemicals with the baby, and we will have the chemicals that pass through the small thin walls of the placentas or rog to the bodies of the baby. So that's one we can call colostrum. Okay, so this one here you can get direct antibody transfer from mother to the baby. And the duration of the protection here in case of active immunization here is take time to develop. Okay, the period of time maybe a month, 2 months, or 3 months, or a week sometimes. But the passive immunization here, when you inject the antibody, so that antibody here can take the action at a certain time. Okay, and the purpose of the active immunization is to protect. Okay, but in case of the passive immunization is for curing or healing process. Okay, this is a process of immunization. The white shape molecules here is antibody, so they bind with the molecule on the surface of a germ. Okay, this is molecules, the specific molecules, and this is the antibody. Okay, so they bind together here, and after that here they can bind into a more complex structures. Okay, for example, this one is complex antibody. This one we call immunoglobulin M. Okay, a specific type of the immunoglobulin. Basically, immunoglobulin here you have immunoglobulin M, immunoglobulin G, immunoglobulin A. Like there's many class, and it depends on. Is quite specific to the different type of a germ. Later, when you learn a lot in biology here, you will know that what is a class of an immunoglobulin here specific to the germ. Okay, maybe in M5. Okay, and the process that the antibody here binds to the bacteria, and it has been eaten by the white blood cells. Here we call opsonization. It's a specific term. So this way here, when you bind the antibody bind with the bacteria like this, so the bacteria cannot be spread throughout your body. It's quite specific to some area. Okay, so it means that the antibody used for stabilize them, so they cannot infect to the other cell else. The next type is the platelet. The platelets or the thrombocyte here is a type of the corpuscles or the blood cells and play the major roles on the blood clotting process ออกการทำให้เลือดแข็งตัว so the size is about only 2 to 4 micrometer or the μm like this is a unit, and it can be produced in the bone marrow. In the classical textbook for you to read here, they say that they produce in the bone marrow. But the new research here, new publication, maybe 2 or 3 recent years here, we found that some kinds of the lung tissue here can produce the platelet too. But this one here, please do not use it for the reference. It's just only the new discovery. Okay. And it takes about 8 to 14 days before they get expiration and get destroyed by the spleen or MAM. Okay, it's produced some kinds of the protein we call thromboplastin. In the news text, sometimes I cannot see the word thromboplastin here, it already cut out. Okay, and use they say that his blood. Sometimes they use the term blood clotting factors. Blood clotting factor like this. Okay, the new one here use this term, and they already cut out the word thromboplastin. But in our talk here, I also mentioned this word because it's shown in some kinds of the examples. The question in some kinds of the examination paper. Okay, so you may need to know this one. And it initiate the blood clotting process. It means that if you lack of the platelet here, you cannot control the bleeding. So it is uncontrolled bleeding or เลือดไหลไม่หยุด Okay. So this one here, as you see this one here, is a step of the controlling. First one, you have injury and the removal of the blood from the blood vessels. So that way it will start the step of clotting. So the first one here, they will produce the enzyme we call prothrombinase. Okay, the prothrombinase here change the thrombin, and they use the calcium and vitamin K. Okay, to change the prothrombin here. When you see the word pro, mean before they turn into a thrombin, we call prothrombin. And you see the word S like this, it is enzyme. Okay, the enzyme. So the enzyme has changed prothrombin into the thrombin. And after that, the thrombin here will change the fibrinogen. So the structures of the fibrinogen is a small fragments of the protein dissolved in the plasma in the liquid part of the blood. But when they act with the thromboplastin, it will change the fibrinogen here into the network we call fibrin thread. So the fibrin thread here will take a trappings of the cells and the protein together. Okay, and it turn to the clot or ริ่มเลือด and after that all this clot here will get enclosed to the wound and stop the bleeding. For example, this one you have injuries of the damaged cells like this. So the first response here, the blood vessel become contract in order to reduce the blood flow. Okay, and after they will turn into the blood platelet plaque. and the platelet plaque here later it will turn into the fibrin clot. Okay, and the fibrin here is protein network to trap the wound. Next one we have blood type. Blood type here we have many types of the blood type. At least we have ABO blood type and RH blood type. And later you can know about the MN blood type, Bombay blood type, and many types of the blood type. But in our talk here we mentioned just only ABO blood type and RH blood type. For the ABO blood type we have 4 variations. The first one is A, B, AB, and O. And for the RH we have 2 subtypes. It is RH positive and RH negative. Okay, we have Bombay and MN, but this one I will not mention about it. The first one is the ABO blood type. For the ABO blood type, here start from A, blood type A. We will. So the question it goes to, what is the chemical used for determining the blood type, antigen or antibody? And this one here we mentioned that the group or the blood type here as a antigen. So what is the antigen? Antigen is a chemicals or the protein on the surface of the blood cells. If the blood cells contain the antigen A, it will be blood type A. But if it contain the antigen B, it will be group B. But if they contain both antigen A and B, like this one and this one, it will be blood type AB. But if they don't have the blood type O, if you don't have any kinds of the antigen, it will be the blood type O. But in opposite to the antigen, you have antibody. Antibody is the one that binds to the antigen. So in a normal case, antigen not supposed to bind with the antigen on the surface of the red blood cells. The question will go to why. If the antigen bind to the antibody in your blood, the blood will become clot and blood cannot flow. So it means that you will get dead. Okay, this way. So if you see the antigen A like this, you will see antibody B, because antigen A and antigen B here never bind together. And if you have antigen B, you will have antibody A this way here. They cannot bind together. And if you have both antigen AB, you don't have any kinds of the antibodies, because if you have one of them, it will bind to the antigen and get clot problems. Okay, and in a blood type O here, you have both type of the antibody. If you see only the word anti here, it means antibody. Okay, and this one is antigen, refer to this one. The same thing. Okay, this one is a blood transfusion pattern. Transfusion means in Thai we use the term การถ่ายเลือดหรือการให้เลือด. Okay, it is the way that the patient had require some kinds of the blood because you have some accident that you lost a lot of the blood volume. Okay, you need blood transfusion. So transfusion pattern here start from O. Okay, you can transfuse O to AB, and you can transfuse O to A and O to B, and even A here to AB and B to AB. So it's AB here we can call the universal recipients. They can receive any kinds of the blood type, and O here is the one that can give to any other blood type. We call O as a universal donor. Okay, ผู้ให้สากลและผู้รับสากล. In case of RH blood type, this RH blood type here, we have only 2 variations, RH positive and RH negative. In case of the antigen that play the roles on this one, it is the antigen D dog, the D, antigen D. If you have the antigen D on the surface of the red blood cells, you will have RH positive. But if you don't have it, you will have RH negative. But this one is not the problem. The problem will go to the antibody. RH blood type is quite different to the ABO blood type. One thing in case of antibody, if you have the RH negative, you're supposed to have the antibody D, right, in the blood. But in case of RH blood type here, first time that you were born and you never get any kinds of RH positive in the body, you don't have antibody D. But when you receive the antigen D into the body, once it will develop the antibody D. Okay, so it means that first step here you cannot detect any antibody D in the body. But when you receive the first time, for example, you receive the blood with from the Rh positive to the Rh negative like this for the first time here. The Rh negative person here will develop the antibody D later. So it means that for the first time here, the blood will not get clot. But if you receive the Rh positive blood to the Rh negative person, for the second time, the blood will become clot. So this is one thing that get different between the ABO blood type. ABO blood type when you get transfuse the blood here for the wrongly from the first time. For example, you transfuse A to the AB like this. The problem will start from the first time. But for the Rh blood type here, you transfuse wrongly once. For the first time here, it will not develop any kinds of problem. But for the second time here, it start to have the problems. คือ Rh blood type ถ้าให้เลือดผิดในครั้งแรกจะไม่เกิดผล มันจะเกิดผลในครั้งที่2 and they lead to the problems. The name of this disease we call erythroblastosis fetalis. Okay, the problem here occur to the occupants with the females contain the Rh negative and the male contain Rh positive. Okay, the problem will go to the baby. Okay, the baby here, if the baby, one of the baby here become Rh positive for the first time. Okay, the Rh positive here will get enter into the bodies of the females, and after that it will the females here, the mother here will produce the Rh. It will produce the antibody D in the body. Okay, so for the first baby here with the Rh positive here show nothing, they are normal. Okay, but for the second time here, that this female here have the second baby with the Rh positive, the antibody D dog here from the bodies of the mother here will destroy the fetus. So when the fetus get born, the fetus here we will have the problems about the blood. The blood cell here become broken, and the bodies of the baby become green. Okay, because the blood cell become broken. So that disease we call erythroblastosis fetalis. Okay, this problems of to the baby. Thank you.